Welcome everyone to this week's edition of the Extracellular Vesicle Club. This is a special edition of the club because we have the ICEV annual meeting coming up, but we wanted to squeeze this one in before the meeting starts. So I'm just going to say a few words about the annual meeting. We have an education day that will be on Wednesday, May 12th this week. And then there are going to be several satellite events over the next few days. Finally, we will have the main meeting from the 18th to the 21st of May, and you can still register for this meeting. So this is the biggest and most comprehensive EV meeting in the world. And it's possible to attend just the education day if you prefer. If you're new to EV studies, want to find out what's going on in the field, what some of the basics are, the education day is a great way to get introduced to that if you're not ready for the full meeting. We also have some satellite events that are going to be free. So for example, there's a collaboration of the Michael J. Fox Foundation and ICEV on neuronal markers of EVs, specifically L1-CAM, and how it can be used or not used, as the case may be, to pull down extracellular vesicles from biofluids to understand the state of the brain. So there's a bit of, I guess, different interpretations and different findings in that area, and we're going to address those head on in a satellite meeting. So today, it's my pleasure to introduce Russell McConnell and Kevin Dooley. They're from Codiak, Codiak BioSciences, and they're going to tell us a little bit about Codiak, and then they're going to go into their recently published paper in molecular therapies. I want to stress here that this is a research presentation, so this is not a sponsor presentation. Rather, they are describing some of the research that they've done at Codiak in engineering extracellular vesicles. And indeed, their constructs, as they'll probably tell us, are available to the community, and they'd be glad to share those with you for your research. So Russell and Kevin, thank you so much for joining today and for presenting your work to the Extracellular Vesicle Club. I'm going to turn the screen sharing over to you now.
Wonderful. Well, thank you very much for having us, Ken. I really appreciate the opportunity to share some of our work. Thanks, Ken, for having us. Really appreciate the opportunity to share some of our work with the community, and also for hosting this, really wonderful series over the past year plus. It's been a great way to stay in touch with, what's going on in EV research. So before we jump into the data, I just wanted to give a one-slide overview of the history of Codiak. So the company was actually founded towards the end of 2015. And lab operations started in the beginning of 2016, which is when myself and Russell joined the company. We've completed a couple of venture raises over the years. Our first headquarters was actually in Tech Square, but we have since moved to our current location in Cambridge in February 2020, just in time to sit down for the pandemic, and completed our IPO in October of 2020. We also initiated 2 phase 1 clinical trials in September of last year, and have recently opened a clinical manufacturing facility to make our engineered EVs. And from the beginning of the company, we were always interested in using engineered extracellular vesicles with very defined therapeutic properties, opposed to relying on any intrinsic therapeutic property of the EV. So in order to do that, we needed to develop new systems for engineering both the surface and the lumen of EVs to display or package different molecules of interest. And this is the work that was recently published in Molecular Therapy. And it's broken down into 2 sections, which is highlighted in the graphical abstract. In the first part of the talk, which is what I'm going to be covering, goes over the purification protocol that we use to isolate EV, as well as the proteomic analysis that we used to identify different scaffold candidates. And in the second part of the talk, which my colleague Russell McConnell will be taking on, and describes how we then use those scaffolds that we identified by proteomics to engineer EVs with different therapeutic cargoes, and ultimately how we tested those both in vitro and in vivo. And so the real backbone of the work is really predicated on both the cell line that we use for EV production, as well as the purification protocol that we use to ultimately isolate the population of EVs that we use. And so this slide here just details the purification protocol. all of the work that was reported in the paper was done with a suspension-adapted HEC-293 cell line. And so this is something that we can grow in chemically defined medium, so there's no serum or anything like that. And we can grow these to very, very high cell densities. And we typically do this anywhere from the 200 mil scale up to 25 liters in wave bioreactors. And at the termination of the cell culture, the cell density is typically between 10 and 15 million cells per mil. So there's a lot more biomass here compared to conventional 2D plastic culture. And so after the cell culture is completed, we remove the cells by centrifugation, filter the conditioned medium, and then treat with a broad spectrum nuclease to digest any extravesicular DNA or RNA species that are in the medium. The media is then concentrated by tangential flow filtration, depending on the size of the production run, and then pelleted by ultracentrifugation to prepare a crude EV pellet. This is then loaded into the bottom of an iodixonol density gradient and fractionated overnight by spinning at 150,000 times G. And when you're working with such high VCDs, you can begin to use visual cues to actually see the different subpopulations that are fractionated on the gradient. And that's shown in the image sort of in the middle of the slide here, where we can see 4 distinct populations after the gradient is completed. And so what we've done in the manuscript is to isolate these different fractions and then characterize them both biochemically and ultrastructurally to get a better understanding of where the EVs are actually fractionating here. And typically what we've done in the past is pull the fractions, ultracentrifuge them again, and then analyze them. But what I wanted to call the group's attention to is this additional 20,000 times G centrifugation step that we needed to add to our purification protocol. And the reason for that is when we imaged that top fraction of the gradient, fraction 1, by 2 different modes, 2 different imaging modalities, we saw 2 different, very different pictures. And so on the left is a conventional TEM grid of fraction 1, and on the right is imaging a similar population just by cryo-TEM. And what we saw was an abundance of this web-like material that coated the entire grid that really wasn't detectable by conventional TEM. So we implemented this 20K spin, which pellets out this web-like material, and what we're left with is a clean fraction of EVs, without any cellular debris. And we characterized both the pellet and the supernatant from that low-speed spin and found the pellet to be primarily composed of actin and actin-binding proteins, which is shown in the proteomics data on the right-hand side of the slide. So in addition to imaging, we also used a number of other techniques to get a better understanding of the different populations that were fractionating on the iodixonol gradient. So we have conventional TEM grids here in the middle of the slide for fractions 1 through 4. And we can see the prevalence of vesicles in F1 and F2. And in F3 and F4, there are detectable vesicles, but they are coated in a blanket of protein. And we can see that more clearly when we analyze these fractions for total protein. You can see in the plots on the right that the fraction 4 contains the majority of the protein as well as the majority of the DNA, which was resistant to the broad spectrum nuclease treatment. We also looked at cholesterol levels, which have been shown previously to be enriched in EV membranes, and we see the majority of the cholesterol associated with fraction 1. And interestingly, when you take these 4 fractions and analyze them on the nanosite, all 4 will register particles, and all 4 will give you size distributions that fall within the described EV range. But we know from the other characterization that we've done that the particles that are registering fraction 4 are probably very different from those in fraction 1, which just goes to show that you do need to extensively characterize the material that you're working with in order to very clearly distinguish EVs from non-EV species. We also used SDS-PAGE and immunoblot analysis to get a better understanding of the different proteins that are enriched in the different fractions. So in the middle is an SDS-PAGE gel loaded by equal total protein from the producer cell lysate, the crude ultracentrifuged pellet that we used to load the iodixonol gradient, and then the 4 fractions. And just by looking at the total protein profile, it's very clear that the material in fraction 1 and 2 is distinct from that in fractions 3 and fractions 4. And when we use immunoblot analysis to look for particular markers, we can see proteins that are involved in EV biogenesis and vesicle trafficking, enriched in fractions 1 and 2, things like syntenin 1, and the tetraspanin, CD9, 81, and 63. Whereas proteins that are bound up in chromatin, things like high mobility group box protein 1 or histone H3, are enriched in the higher density fraction 4. And there's one particular protein I want to call the group's attention to just because it was really useful for us as we were developing our purification protocol. And that's galectin-3 binding protein, or LGAL-S3BP here. We can see that it's very enriched in the UC pellet and predominantly fractionates in fraction 4. And this was something that we used as a marker for contamination, essentially, for EVs derived from HEC293 producer cells. And we looked at a bunch of different methods for isolating EVs, things like PEG precipitation or PEG coupled with size exclusion chromatography, which is shown in the protein gel here, as well as ultracentrifugation. And you can see by the blots down below for galectin-3 binding protein, all of these methods, PEG, PEG plus SEC, and ultracentrifugation alone, result in an enrichment of galectin-3 binding protein. And we really needed to use the iodixanol fractionation in order to deplete that contaminant from our EV population. And interestingly, there's a bit of literature on this protein, which is also called MAC2 binding protein, and it forms oligomeric protein structures that form these rings, which can be imaged by TEM. And I know there's no scale bar on here, but these are about 35 to 40 nanometers, which I think could be easily confused with EVs by just looking at TEM grids. Now, we also used LCMS and proteomic analysis to get a more granular picture of the proteins that are enriched in these different fractions. And that's shown in the heat map on the left. So we're showing here a subset of that analysis and highlighting proteins that are enriched in fraction 1 in the top portion of the heat map, and those enriched in fraction 4 in the bottom portion. And if you look at the specific proteins, you can see they're very different. Fraction 4 is primarily composed of secreted extracellular matrix constituents and histones, which are probably responsible for the high amounts of DNA that are still present in fraction 4 after nuclease treatment. And if we look at what's enriched in fraction 1, we can see proteins that are again involved in vesicle biogenesis, things like ALEX, syntenin 1, lactoherin. But we also found a number of proteins that hadn't been previously well characterized as being specific to EVs. And there are two families of proteins here. One is the EWI subfamily of the immunoglobulin superfamily, and the two members are PTGFRN and IGSF8, shown with the pink arrows here. And then we also saw a number of proteins from the MARCS protein family enriched in fraction 1, including MARCS, MARCSL1, and BASP1. And so these were the candidates that we had identified from the proteomic analysis. I have a schematic in the middle of the slide here, which just shows the differences in topology of the scaffolds that we identified, as well as tetraspanins, which have probably been used most widely as a mechanism for engineering EVs. So the MARCS protein family falls into this lipid anchor engineering scaffold. All three of these proteins contain an N-terminal meristylation modification, which is anchored into the inner leaflet of the EV membrane. And then PTGFRN and IGSF8 are single-pass transmembrane proteins, which are similar to sequences like LAMP2B or p-display. And now to specifically assess the capacity of each of these candidate scaffolds to direct fusion proteins into EVs, we used flag tag GFP as a surrogate cargo. So we made genetic fusions of each scaffold to GFP and then stably transfected an exosome producer cell. So this is the HEC293 suspension cell that we described earlier. And all of the work that's in the manuscript is from stably selected cell pools. We found that there's a lot of variability associated with transient transfection, as well as some other potential artifacts, which can make downstream analysis quite challenging. So everything is from a stably selected producer cell. And everything is also purified using the iodixanol gradient that we just walked through. And now, as a way of directly comparing the efficiency by which these scaffolds can direct GFP into EVs, we first looked at the producer cell. And we wanted to make sure that all of the producer cells could indeed make the fusion protein at the cellular level. And we're looking at flow cytometry data on the left-hand portion of the slide. I'm looking at all the different scaffolds as well as a cytosolic GFP control, which has no membrane targeting sequence. You can see the cytosolic GFP results in about 10-fold higher GFP than the rest of the scaffolds. But for the most part, they're all about the same, with the exception of LAMP2B and P display are a little bit lower. But then when we look at the levels of GFP in the EVs from these producer cells, we see a pretty wide spread. And so this data was generated using quantitative ELISA coupled with nanoparticle tracking analysis to generate, to quantitate the number of GFP molecules per EV. And what we're showing here is that the scaffolds that we identified by proteomic analysis resulted in the highest levels of GFP per exosome compared to previously described scaffolds. And the last piece of data I wanted to share with the group is actually a bit of deeper characterization on those EVs themselves using nano FCM to look at the single vesicle level. We were curious if we were isolating different subpopulations, some of which would be highly enriched and some not. And so we looked here, looking at GFP expression on the x-axis and normalized count on the y-axis. We see really nice monodispersed populations of GFP positive EVs for the 4 scaffolds that we identified by proteomics. So now I'm going to pass it over to my colleague, Russell McConnell, who's going to walk us through how we use these scaffolds to actually engineer EVs.
All right, great. Thanks, Kevin. So when we were looking at these, we first took an approach where we wanted to look at overexpression of the full-length proteins, and then really dive into which portions of the proteins are required for very high-level exosome expression. And so on the left here is just a schematic of the PTGFRN truncations that we tested. So PTGFRN has 6 Ig domains on the outside of the on its extracellular portion, and basically we just did a serial truncation, essentially just removing 1 Ig domain at a time. And what you can see is that there's something a little funny going on, where as you truncate off these, you actually lose quite a bit of expression, but then the very shortest truncations, so the 687 here, actually expresses at a pretty decent level. So some work that I'm not going to go into now, but we go into in the paper quite a bit, is actually looking at what's going on with these longer truncations, and it's actually destabilization of the protein, but the shorter truncation actually loads quite well. And really, I think highlighting just how well PTGFRN localizes to exosomes. If you look, and this is just a SDS-PAGE protein gel. So this is a wild type exosome, and this is the PTGFRN band here. If you overexpress PTGFRN, you can actually get this massive band of protein here, where it's by far the most abundant protein that you can see in the protein gel. And if you look at cryo-EM images of a wild type exosome and compare that to the overexpression, you can actually see this like protein corona kind of surrounding the vesicle in the PTGFRN overexpressing exosome. And we believe these are actually the PTGFRN molecules sticking out from the exosome surface. So you can express PTGFRN at extremely high levels on these vesicles. So what can you do with this protein? So we actually use this system to display a number of different types of proteins on the surface of exosomes. So this ranged from a secreted molecule in the case of IL-7, all the way up to trimeric protein like CD40 ligand, single chain Fabs. So this is an anti-CD3 single chain Fab that's displayed on the N-terminus here, as well as IL-12. So I'll go into a little bit more detail in the next slide on what we saw with these constructs. So the interesting thing here is that you're actually able to confer biological activity onto these EVs by generating these fusion proteins. But we didn't see that all of these sort of worked equally well when you compare them with some other controls. So this data is highlighting these four different examples, and they're using different assays. So generally speaking, we're either looking at cells isolated from human blood or cells isolated from mouse spleens. So these are all assays that we're running using primary cells. So in the case of IL-7, if you look, in all of these, we actually use two different versions of PTGFRN, either the full length shown in blue, or that very short deletion construct that still worked quite well, and that's shown in red. So if we compare this versus P-display, which is the PDGFR transmembrane region, which we used as a control for a lot of these experiments. If you look in the case of IL-7, you can see that the PTGFRN fusions actually did quite a bit better than the P-display version. But if you compare it to recombinant protein shown here, actually all of the exosome, the vesicle versions of IL-7 didn't really work as well as just the purified recombinant protein. So this actually contrasted quite a bit with what we saw with some of the other constructs. So in the case of CD40 ligand, again, the PTGFRN fusions worked quite well relative to the P-display version. But in contrast to IL-7, the CD40 ligand actually worked quite a bit better. So they had lower EC50s in the case of the short fusion, as well as the full-length PTGFRN fusion, when you compared it to either the recombinant protein or the P-display version of this. And again, you see something similar when you look at the activity of the anti-CD3 single-chain Fab. So the full-length version of PTGFRN actually did a better job of displaying this than the short version, and they both seem to be pretty active, relative to the recombinant protein. And in the case of IL-12 fusion, we saw similar activity between the two PTGFRN fusions, and we also saw pretty comparable activity to recombinant IL-12. And this is really just looking at the interferon gamma secreted from PBMCs following stimulation with an anti-CD3 antibody. So I'd like to dive into this IL-12 data a little bit better, a little bit deeper. And what you can see is that when we treated mice that had tumors implanted in them, when we looked at the response, so here we're just looking at the full-length IL-12 PTGFRN fusions, we saw that at the 100 nanogram dose, these actually did a lot better. So the tumors were much smaller than either with the recombinant protein or PBS control group. And when you increase this to 200 nanograms, the recombinant protein kind of caught up to the IL-12, the exo IL-12. But, and in both cases, they were doing better than the PBS treated controls. And if you look at the survival curves associated with these, you can see that, in both cases, the exo IL-12 and the blue group here did better than either the recombinant protein or the PBS controls. So all of this data together was showing us that this IL-12 PTGFRN fusion protein that was displayed on the EVs was really having a potent anti-tumor effect in these mice. And I just wanted to plug this paper that was recently published from some folks here at Codiak that goes into this result in a lot more detail, but they're using here IL-12 fused to the PTGFRN molecule that we're describing. So that was all work where we were displaying proteins on the surface of the EVs. So we next wanted to look and see what was happening with the Marx family proteins and what was required in those to localize proteins and protein fusions to EVs. So the Marx family proteins that we identified, so Marx, L1, and BAS1, share a number of common structural features. So they're mericylated on their aminoterminus, and in the case of Marx and Marx L1, have a polybasic region that sits sort of in the middle of the protein. BAS1 has a slightly different organization and has a polybasic region that's proximal to the aminoterminus. So we basically expressed either full-length versions of these or just the amino terminal 30 amino acids of these proteins. So in the case of Marks and Marks L1, this construct just has the myrcellation site, whereas in the case of BAS1, it has the myrcellation site and the polybasic sequence. So similar to what we did with the PTGR fusions, we looked at the producing cells by flow cytometry to examine GFP expression just to see if they were all making the protein. And we saw extremely similar levels across the board for all of these constructs. When we looked at the purified exosomes, and again use a quantitative ELISA to determine GFP levels and normalize that to the particle counts, what you can see is the full-length proteins, all three of them express pretty well, and they localize to exosomes decently. We saw something very different when we looked at the amino-terminal truncations of these proteins, however. So both Marks and Marks L1, when you're using just the first 30 amino acids of these two proteins, had much lower expression than BAS1, which had a very high level comparable to what we saw with the full-length proteins. So we did a little bit further analysis and truncated this down further. And shown on the left here is actually Kamasi stained protein gel, as well as the corresponding anti-GFP Western blot. And what you can see is we did a serial truncation from the 1 through 30 sequence all the way down to the first 3 amino acids. And what you'll notice is that you have very strong localization for all of these constructs up through 1 through 9. And then when you chop off and you just have the first 6 amino acids, you lose this pretty strongly. And if we dive in a little bit deeper, we were able to trim this down through the first 8 amino acids. And you start to see some loss when you get down to just the first 7. So the consensus sequence that seemed to work the best was this 1 through 8, containing the sequence NGG, KLS, KK. And you can see there's a couple additional lysine residues here in the positions 9 and 10. So for most of the work that I'm showing from now on, we'll actually be looking at the first 10 amino acids on this. So in addition to this polybasic region, which seems to be required for the localization, we also wanted to see if the myrcellation was required. In work that's been long established, the way that proteins get myrcellated is the initiator methionine gets cleaved off by methionine aminopeptidase, and then the exposed alpha carbon on the glycine in position 2 has the myristoyl group attached to it. So what we did was mutated either this second glycine, which is the myrcellation acceptor site, or the third the glycine in position 3, which shouldn't have any effect on myrcellation. And what we see is that the glycine to alanine mutation in position 2 basically eliminates EV localization, whereas mutating the glycine in position 3 has no effect. And if you make the double mutation, you of course abrogate binding here as well. So this is the the protein gel and the Western blot data for all this localization. But you could actually kind of tell what was working and wasn't working just by looking at the gradients. So at the right here is actually an image of the iodixinol gradients. And if you look at the band for the EV proteins, you can actually see that the 1 through 9 sequence and the G3A sequences are actually green. So you can actually see the GFP by eye for these, whereas the other bands where you don't have strong localization of the GFP construct have a more whitish color to them. So this is a really very pronounced effect that's readily apparent when you're looking at the samples themselves. So what can you do with this? So we've actually used this construct to load all sorts of different things into EVs. So, the work I was just showing was GFP, but we've also used this to load, chemically induced dimerization systems. We've actually used it to load transmembrane proteins as well, affinity reagents, so camelid nanobodies, RNA binding proteins, Cas9. And this is all pretty impressive for this short little 1 through 10 peptide sequence. So all the crystal structures here are roughly to scale. So this tiny little peptide here is capable of redirecting all of these different proteins into EVs just by fusing it to their amino terminus. So I'm going to talk a little bit more about this particular protein, ovalbumin, which we actually use as a classic antigen for vaccine formulations. So what we're doing here is actually loading the vesicles with ovalbumin, as well as a cyclic dinucleotide STING agonist. So again, I'm going to plug this another paper that recently came out from the group here that was looking at using these cyclic dinucleotides to basically activate the immune system. And here they're using them as a way of basically melting tumors. So if you'd like more details about the STING agonist and how it works, you can see this this recently published paper. But in this context here, we're actually using the STING agonist as an adjuvant. So what we're going to be showing in the next couple slides is ovalbumin either as a free protein or loaded inside a vesicle. And we administered this in a couple of different ways. So one that we call exovac is actually the antigen plus the cyclic dinucleotide loaded in the same population of vesicles. We also tested if you just have the antigen inside a vesicle and you co-administer the cyclic dinucleotide not associated with the vesicles. And then finally, we looked at free protein and free protein plus the cyclic dinucleotide. So what you can see from the data presented in the graphs on the right, so we looked at this a couple of different ways. So in this panel, we're looking actually at the tetramer staining for MHC class 1. So this is basically detecting effector memory T cells that are loaded with the ova peptide synfecal on MHC class 1, and we're detecting this by flow cytometry. And what you can see very clearly is that the exovac formulation was really the only thing that elicited a very strong immune response. So actually, if you give the ovalbumin inside of an exosome with a STING agonist, but the STING agonist isn't actually associated with the vesicles, you fail to elicit this response. And you also don't see a very strong response if you administer either free protein plus the adjuvant, or just the antigen inside the vesicle alone. And I should mention here also that we're looking at both effector memory T cells from the lung. So these were all administered intranasally. So this is looking at a local immune response, as well as looking at T cells from the spleen. So this would be, both local and a systemic immune response that we're able to elicit with the exovac formulation. So you also see something very similar when you look by ELISpot. So here we're looking at interferon gamma secretion from splenocytes, and we're looking both for the CD8 and CD4 peptides from the ovalbumin. So in both cases, we're seeing a very strong response in the exovac administration, but much less response from the other conditions. So the other question that we had is, all of those experiments were done with intranasal administration. Are other routes of administration also viable? So basically, we took the exovac formulation and administered either intravenously, intranasally, or subcutaneously, and in all cases saw a really nice, robust response. And for comparison, we're looking at subcutaneous administration of OVA plus a more traditional adjuvant, Ativax, which is used in vaccines for a number of viruses. So compared to the sort of traditional formulation of protein plus adjuvant, we're actually getting a much better response with the exovac. So just to summarize, we're looking here at some new scaffolds that we identified by using the high purity EV isolation that Kevin outlined at the beginning of the talk. And this was really critical to be able to separate things that are truly vesicle associated from other things that were co-purifying with the vesicles using some of the preparation methods that we initially looked at. So these new scaffolds can be used to load diverse types of proteins, and you can also load multiple cargoes simultaneously, either by just expressing two different things fused to PTGFRN, for instance, or you can display things on the outside of the exosomes, the vesicles using PTGFRN, or on the inside using BASP1. As we showed with the vaccine data, you can actually combine these protein engineering methods with other loading methods. So in the case that we presented here, is a small molecule, so a cyclic dinucleotide sting agonist that was added to these engineered vesicles. So we think that the scaffold discovery workflow that we've outlined here, of course, is readily adaptable to other types of vesicles. So you can look at vesicles from other cell lines, from tissues, from body fluids, lots of different sources. But really, keeping the emphasis on high purity vesicle purification and really rigorous validation of your capability of isolating the vesicles away from other co-purifying molecules. So we've tested the particularly PTGFRN and BASP1 fusion proteins in some other cell lines, and they've worked in the cell lines that we've tested, but we'd really like to see these tested in other EV systems. And it is worth mentioning that we have isolated vesicles from a number of different cell lines, and we've detected these protein families in pretty much every vesicle type that we've examined so far. So in this table, you can see there's a number of different cell lines, and they all have, at least one member of the EWI protein family and at least one member of the Marx protein family. And in most cases, they actually have quite a few of these proteins of interest. And then last but certainly not least, I will mention that there are currently 2 clinical trials underway that utilize these engineered vesicles. So EXO-IL-12 is currently being evaluated for early stage cutaneous T-cell lymphoma, and EXO-STING, which is the PTGFRN overexpressing exosome loaded with the small molecule STING agonist, is currently being investigated for use in solid tumors. And with that, I would just like to thank the many, many folks who have contributed to this work, and I would be happy to take any questions.
Well, thank you very much, Russell, and thank you, Kevin, for presenting this exciting work to us and telling us about some of the things that you're doing with these engineered EVs as well. So we will now open this up for discussion, and I'm going to I'm going to ask for specific people to please unmute yourselves. So when I call your name, please go ahead and do that, and you can ask your question. So we will start with Rajagopal Ramesh, who has a question about the Galectin-3 protein. Please go ahead, Rajagopal.
Hi, thanks for the excellent talk. My question was, so the Galectin-3 expression, is it unique only to the 293 cells, or have you tested in any other cell line? Because at least in the preclinical studies, other cell lines are also used as producer cells. So for EVs, so do you have any idea about it?
Yeah, thanks for the question. Unfortunately, we haven't really tested for the presence of Galectin-3 binding protein in other EV production cell lines, just in HEC-293.
Thanks.
Great. So we next have a question from Guido David, and it appears that you're not on, but I think I did see your name still here. So Guido, are you there? I think I'll go ahead and just ask this question then. It's about gamma secretase. So does the truncation of PTGFRN cause it to become a constitutive substrate of the gamma secretases, which would include the presenilins?
I don't think that's actually what's going on here. So some of the data we weren't able to present in the talk is actually looking at the producer cell lines that we use to make those truncations. And if you look at the GFP fluorescence, it's pretty constant across all of them. So what I actually think might be happening is they're getting hung up either in the ER or some organelle and not actually making it to the EV membrane, would be my guess. But that would require further investigation. But I don't think they're getting chewed up by gamma secretase.
Great, thank you. Next is John Sperduto. So John has several questions about the processing procedures.
Hi, yeah. First, really good presentation. Really welcome to see the thorough characterization that what you're using is actually EVs. Definitely welcome. So yeah, my questions were foremost, so something that people are starting to notice, and I've noticed myself having done UC and TFF, is UC seems to deflate the EVs, in a sense, whereas TFF, if you look at the TEM, they tend to be much more rounded. Do you think that that is what's happening here? Or do you have plans to switch to TFF or perhaps another isolation method? Because obviously the iodixonol gradient is not a scalable bioprocessing method like others are. And then the second question is just an idea. What do you think that fibrillar structure is? Do you think that that's artificial as a function of the UC, or do you think it's something inherent to how EVs interact with soluble fibrillar ECM? And then the third one, which is more just a curiosity, do you think that any of this PTGRFN loading would vary between adherent and suspension cell lines?
I think maybe I'll take the first two, and Kevin can take the last question there. One, the deflating vesicle question, I think it really depends on how you're looking at these the sample. So if you're looking by negative stain EM, of course you're putting them into a vacuum, and some of them deflate, and then you get sort of like a flattened football shape, right? But if you look by cryo-EM, where you're just freezing the sample in place and then examining it, we haven't really seen any effects like you're describing with the UC purified material. So the vesicles appear to be intact and roughly spherical. So I don't think that that's necessarily an issue with the purification, maybe more of an issue with the visualization method. We have published I think in a few places now, but for large scale applications, we're not using the ultracentrifugation for EV purification. So we have come up with a scalable chromatographic method to purify EVs, and that's what's being used for clinical scale production. So the fibrillar material, we're not exactly sure where that's coming from, but it may be a consequence of using a high density suspension cell culture. So it's definitely, all the characterization that we've done has been showing that it's basically an actin-based structure. So it's an actin protein and a lot of actin binding proteins that are associated with it. And it's not really associated with the vesicles, because you can actually separate it from the vesicles quite easily using this low speed spin that Kevin outlined. So we're not exactly sure where it's coming from, but it's probably coming from dead cells that are basically releasing their contents out into the medium. And it's not exactly clear why you only see it, why it's making it through the purification process, because there is a lower speed spin prior to the ultra speed spin that should theoretically get rid of it. But it makes it through that step somehow. So, we see it very reproducibly in the, that fraction 1 material when we pull the iodixonol gradient fractions, but we're not exactly sure where it's coming from.
Great. And then for the last question you asked here about differences in loading between suspension versus adherent, unfortunately, I don't have a great answer for you. I think this is something that is going to need to be tested empirically. And as Ken mentioned at the top of the hour, we're working on getting these plasmids available just so people in the community can try this out in different systems. My suspicion is it is going to work. We tried a number of different HEC-293 cell lines. Granted, they are grown in suspension, but it's not specific to the one cell line that we've tested here.
Awesome. No, yeah, all makes sense. Awesome presentation, awesome work. Looking forward to what's coming.
Thanks for the questions, John. And again, just to emphasize, as Kevin said, these plasmids are being made available. So feel free to reach out to Kevin and Russell if you're interested in trying this system, or these systems, I should say. So the next, we have a couple of questions here about the mouse model from Maria Elisa Vasquez, and then also a follow-up from Patrick James. So if the two of you want to go ahead and ask your questions in sequence, please. So perhaps Maria is having a problem with her microphone, so I'll just go ahead and ask this. Question about the dose. So 100 versus 200 nanograms in tumor therapy, is that total protein in EVs, or is that a specific protein that you measured?
That is the IL-12 dose.
So that's the answer then. That's the dose related to IL-12. And Patrick, go ahead.
Yeah, so it's following on from that. So with the IL-12 PTGRFN, how was the mice dosed? Was it intratumorally, IV?
Yep, so all of them were dosed intratumorally in those studies.
Thank you.
And Patrick, you had another question too, so why don't you go ahead and ask that?
Awesome. Yeah. So you mentioned at the start that you did a lot of your work with cell pools. I was wondering whether have you, I assume you've done some limiting dilution, so you've gone for like monoclonality. Have you noticed, so what kind of variation on EEV yields have you observed?
That's a good question. I don't think I have a great answer for you, unfortunately. I think things are pretty consistent from cell pools to individual clones, but again, can't comment directly, unfortunately.
Thank you.
All right. So next questions come from Kyle Schuler.
Yeah, thank you for the great talk. I just had two questions regarding the PTGFRM platform, and if you've tried to utilize this to adhere specific targeting proteins or proteins with known targeting properties for specific cells or tissues to the outside of the EVs. And then my second question was just regarding your transfection methods. I think in the paper you mentioned neon electroporation and a transporter 5 reagent. I was just wondering if you've seen any particular method to be the most effective, and maybe how that compares to other methods like lentivirus, stable transfections, things of that nature.
Yeah, I think I can hop in here. So yes, absolutely. To your first question, we have thought about and are currently working on appending all different sorts of targeting proteins to the EV surface using PTGFRN. It's a large body of work that we're currently focused on. And in a sense, I mean, appending like the anti-CD3 single chain Fab is in a sense a targeting ligand. So yep, that's something we're actively pursuing. And then in terms of the transfection method, we haven't noticed really any difference in terms of transfection efficiency or protein expression using either the neon electroporation or transporter 5 method. But we haven't compared that directly to lentiviral transduction. We stuck with transporter 5 and electroporation really just for ease of use. It's very quick to do. But again, yeah, haven't noticed any differences between the two that we routinely use.
Okay, yeah, that's great to know. Yeah, thank you so much.
And I'll just add quickly too that, as Kevin mentioned, we're also putting these through stable selection. So, it's not a transient transfection. So small differences in transfection efficiency kind of come out in the wash after the selection.
Right. Yeah, that makes sense. Thank you.
So our next question, which I will just ask myself, I think I'll ask the next two myself, but the questioners are welcome to come on too if they have follow-ups. How do you inactivate benzonase? Or do you?
We don't. Yeah, so the benzonase activity is removed through the sequential steps, including density gradient fractionation. It's something that we've tested, and we don't see any residual benzonase activity in fraction 1 after we pull that band and put it through the subsequent purification.
Great. Okay, so that was a question from Carla Pereira. So thank you, Carla, for the question. And next from Artem Zhivolozhny. Have you tested the EV samples for negative controls or depleted controls like apolipoproteins, GM130, histones? I think you had some of those in there, didn't you?
Yeah, so we blotted specifically for a couple of different histones, and we don't see any of that in fraction 1. We see an accumulation of those types of proteins in fraction 4, which was also shown in the proteomic table. We didn't specifically look for GM130 or apolipoproteins by Western blotting, but we don't see any of those proteins by proteomic analysis.
Great. Okay. Thanks for the question, Artem. We will next go to Genevieve Bart. When you do you want to ask your question about the BASP1?
Well, actually, it's hello, very interesting. It's not exactly just about BAS1, but I was just wondering if the expression of the protein in the EVs is correlated with expression in the cells, because you showed in your last table that some of the cells you tested didn't express. I just wrote BAS1, for instance, that was not expressed in the EVs from K562 cells. But is it expressed in the cells, and is the level similar to what you see in hex cells?
Yeah, thanks for the question. So you're absolutely right. The cases where we see where we don't see these proteins localizing into the vesicles, the cells aren't expressing the protein. So I think it's more of an issue with protein expression and not really with localizing these proteins to the vesicles themselves.
Okay, so you can predict, at least for those ones, you can predict if the cells express them, the EVs will have them.
Yes, in the limited number of cases that we have, that has been true.
Thank you.
Thank you. Next is Marcello Clarici.
Hi. Yeah, I'm not sure if you have partially answered this in a previous question, but your actin-related proteins, you seem to get rid of them in the 20,000 G centrifugation step. But don't you expect somehow to get rid of them also in your flotation step? So I just want to know if you have some data about this, or if maybe you suspect they are somehow associated to the EVs. Thank you.
Yeah, that's a great question. Yeah, if it was just actin and actin binding proteins, you would expect them to be very dense and to not wind up in that fraction 1. And so the way that we're running the gradient as well is that we're mixing our sample with the iodixanol and loading it at the bottom of the gradient, and then floating the EVs up to fraction 1. So you would expect very little material in that fraction 1 that doesn't have a significant membrane component to it. So we actually don't know if there's cellular membranes associated with the actin and actin binding proteins that are present in that sort of what we call web material. But that would be my guess as to why it's floating up to the top. And when you look at it higher magnification in the cryo-EM images, you don't necessarily see just actin fibers. So it's probably cellular membranes associated with the actin fibers, and that's what's causing it to float to the top of the gradient.
Okay, we have, I see 3 more questions, so we'll try to get to all of these. And Jay, hold on, because I see your question, but the other 2 are sort of related to each other. So I'm going to go first to Jagdeep Sandhu. You have a question about microRNAs.
Hello, excellent talk. I really learned a lot. I have a very simple question actually. So these EVs, they're not just nothing, right? Not inert. So they have a lot of microRNAs, proteins, and other things. So what could be the effect of these on the recipient cells?
Yeah, that's a. Oh, do you want to take it, Russell?
Go for it, Kevin.
No, yeah, that's a great question. I guess there was a spirited debate at the previous session of the EV Journal Club on the microRNA content of EVs. But I'll save my personal commentary on that. And but just for your question, in all of the activity data that we presented in the manuscript, we always include an unengineered EV control at the same dose that we're giving the engineered version. And we haven't measured any detectable phenotype of the unengineered HEC293EV, whether that's mediated by a protein, a lipid, or a microRNA, we can't say, because there's really no measurable impact of treatment with EVs alone.
But when you say no measurable impact, so what are we measuring? Like, how do we know what kind of side effect it might have, right? So if we don't measure, we don't know, right? But if we measure, what do we measure?
Sure. So in the case of the different engineered constructs that we tested, we looked for particular activation markers associated with those proteins. But we've also looked at things like cell growth, cell viability, things like that, and haven't been able to really detect any sort of changes in biology associated with treatment with the unengineered EVs. It's kind of hard to look for something when you don't know what it is you're exactly looking for.
Okay, thank you. At least it's something you're looking at. They're not causing a lot of proliferation by themselves. They're not becoming tumorigenic or something like that, right?
Right.
Okay, thank you
Yeah,
so
so
much.
that is indeed the next question that we had from Yoon Jae Jones. So Yoon Jae, do you want to ask this too?
Yeah, thank you for your exciting presentation. But my question is following with objective questions. But yeah, actually, because of the safety issue, the tumorigenicity or immunogenicity is very important for clinical trials. So, and HEK293 cells are known to tumorigenic. So I think their EVs also have tumorigenic potential. So did you remove their proteins or microRNAs from HEK EVs, or maybe you think their effects are marginal, right? So what do you think of this?
You want to take it, Russell, or you want me to jump in?
Sure. Yeah, so as Kevin mentioned, we haven't seen any effects of these vesicles when we administer them to either cells in vitro. We've done a number of animal studies where we're also not seeing any tumorigenic effects of these vesicles, and that's in everything from mice up through a number of different non-human primate models that we've looked at. So we're not overly concerned with tumorigenic potential of these, but of course it's something that we're of course keeping an eye out for. But we haven't seen any signs of that in any of our either preclinical data, and I don't think we've released any of our clinical data with these just yet. But that's of course something that we're going to be paying close attention to in the phase 1 trials.
So I think I would actually recommend that these be tested in some iPSC derived cells, because at least those are human. As we know, like many things we don't observe in rat and mice, we do see some effects in the human cells. So maybe it would be nice to actually do in some kind of iPSC cells.
Yes, it's a great suggestion.
Very good. All right, and we'll come back then to Jay. So Migara Jayasingh, you've I think that Kevin and Russell have shown us that they're able to put a lot of proteins in, but Jay has a question about the cargo.
Hi, thanks for a great talk. It was amazing stuff here. I've read your papers on the sting agonists and IL-12, and the data is amazing. I was just wondering if, I think a couple of years back at AACR, you all presented some work on the mRNA vaccine where you showed data, or I'm not sure if it's just a schematic where you load mRNA. Do you have any data or any direction where you all have tried to load the nucleic acid cargoes, either parental cell transfection or exogenously, or either through lipid insertion or any other methods?
Yeah, absolutely. And so Russell actually briefly touched on this, but using the BASP1 sequence to load RNA binding proteins is a method that we've explored for specifically loading RNAs that contain a cognate hairpin sequence into the lumen of exosomes. And we've also done quite a bit of work exogenously loading different oligonucleotide sequences onto the surface of the EV using things like lipid anchors.
I see. Thank you very much. So you do see significant functional, enough to get a functional effect, right?
Depending on which avenue we've used for loading, the activity is a bit variable, but that's something that we're continuing to develop and work on here.
I see. Thank you very much.
So I have a brief follow-up on that. You mentioned, Kevin, that you have some personal ideas on, or personal views on microRNAs and EVs. I'm wondering if there's any, if you can say anything about what would happen if you had, let's say, an Argonaute protein that was now engineered to be attached to one of these scaffolds. It would obviously be in very close proximity to the membrane. And I'm wondering if the activity or the cargo of that specific RNA binding protein might be different from one that is essentially cytoplasmic, not necessarily in proximity to the plasma membrane or any membrane.
Absolutely, I think that's a really good idea, and I think the Jeppesen work that came out in Cell in 2019 did a very nice job, I think, in my opinion, debunking the whole Argonaute loading hypothesis, but specifically tagging them with sequences that would localize them to EV membranes might completely, turn that on its head. That's a great idea.
Okay, everybody, I hope you heard that. Everyone get out there and do the work. And I see we have one final question from Rupadya. Go ahead and ask your question, and I think this will be the last one for our session. All right, let me just ask this then. Is it possible to release the protein of interest that's attached to BASP1? So you load it, but then maybe you don't want it right there. You want to release it. Is that something that can be done?
So I think either BASP1 or PTGFRN can be used very nicely as sort of platforms. So if you have an inducible loading system, you can certainly employ that. And there's been a number of those used in the literature. But I'd like also to mention and highlight there's actually a whole body of work around BASP1. So it was originally identified as a cofactor for transcriptional regulator Wilms tumor 1, WT1. And there's this beautiful body of work showing that actually BASP1 can shuttle between the membrane and the nucleus in a calmodulin regulated fashion. So in a very highly unusual binding mode, calmodulin actually binds to the myristoyl group on BASP1, sort of covering up that hydrophobic sequence, and that allows the whole protein to translocate into the nucleus. So this is something that we haven't, really examined in depth, but it's possible that this system would be, allow proteins to come on and off of the membrane. And this whole system is regulated through PKC phosphorylation of the serine in position 6 in BASP1. So like I said, there's an extensive literature on this already, but not in the context of vesicles. So, if somebody wants to look at that, please do. I think it's fascinating, but it's not something that we followed up on.
Very cool. Well, I think we've raised some questions today too, in addition to hearing about the exciting work that you've already done. So I think that's a very nice use of our time, to come up with some ideas too. So Kevin and Russell, thank you so much for joining us today. We really appreciate your taking the time to share your research with us. We wish you the best as you continue the work that you're doing. And thanks to everybody for joining today. Thanks for the questions. Thanks for the participation. I hope to see many of you at the ISEV 2021 meeting starting on Wednesday this week. And we'll be back to regular Journal Club sessions then at the end of May. So if anybody would like to sign up to give a talk or to moderate a session, just feel free to reach out to me and let me know. So thanks again, everybody. Take care, stay well, and we'll talk to you soon. Bye now.
Thanks, Ken. Take care.